Hello and welcome to the fifth part in our series on vision in physiological psychology. We have one more after this, which will be actually lecture number 12, which is color vision. The final step here in talking about physiology of vision is to talk about the visual cortex. I wanted to first review a little bit about receptive fields, then we'll talk about the organization of the visual cortex, or the striate cortex. We'll talk about feature detectors and their role in perception, mapping the visual cortex, and then we'll finally finish up talking about the what versus where pathways. It's just a reminder that the receptive field is an area of the retina that affects the firing rate of a given neuron in any circuit. So receptive fields are determined by monitoring a single cell responses. So when we start talking about the visual cortex, we're going to be talking about the receptive field and the specific shape of the receptive field and the orientation of the receptive field, as well as the location of the receptive field. So if we present a stimulus to the retina, the response of that cell is then measured by an electrode or other process, primarily single cell electrode recording. That's how this work has primarily been done. And I wanted to review quickly the idea of center surround receptive fields because those become important as we start talking about simple cells in particular. So you recall from our last lecture, we might have a receptive field that looks like this annulus with an on center, or it could have an off center. So if we shine a light in the center, it's going to excite the cell and increase its firing rate in the on center field. If we put it in the center of an off center field, it's going to reduce the firing rate of that neuron. If we illuminate the annulus, it's going to reduce the firing rate of this on center. It's going to increase the firing rate of this off center. And then if we do diffuse illumination, it's really not going to change the firing rate all that much. Now this is going to become important when we start talking about how simple cells start recognizing features of objects. So the visual cortex is comprised of 6 layers arranged in columns. There are 3 different types of simple cell or cells we're going to talk about in the visual cortex. These are the ones we'll talk about in this course. These are in the primary visual cortex. So to give you an idea of what this looks like, so we're going to talk about simple cells, complex cells, and end-stopped cells. So this is essentially how Hubel and Vazel conducted their research. We have a cat here staring at a screen with an electrode and a single neuron, and then we see which type of light stimulus that neuron likes to respond to. So in this case, this vertical bar is what that neuron responds best to. It responds okay to closely related stimuli, but the further we get away, the less that neuron responds. So the first of the cells we'll talk about in the visual cortex are simple cells. These are side-by-side excitatory and inhibitory receptive fields. So you can see, for example, if we present a light in this area, we're going to get maximal stimulation. Now if we present it this direction, we'll get some more stimulation than inhibition, but if we present something horizontally, we're going to get as much excitation as we do inhibition, so we won't get any response. Now, one of the things I mentioned is that this type of simple cell receptive field is due to center surround antagonism. And essentially what has happened is we get input from multiple center surround antagonism cells such that we get maximal, when we get these overlapping receptive fields, we actually create this kind of bar shape that this simple cell will respond to. And these have varying orientations based on their inputs. So these cells respond best or least to a bar of a light in a specific orientation. So these are bars of light in specific orientations. And this is how we start to extract visual features and recognize items in our environment is through this particular process. So we can see we get neurons that respond particularly to stimuli in particular orientations. Now, these cells again respond best or least to a bar of light in a specific orientation, and this results in what we call an orientation tuning curve, whereby In this particular case, this would be a vertical simple cell, so it responds best at this perfect vertical, and the further we get away from that, the less the cell responds, until eventually we get to the point where the cell doesn't respond at all. So it will respond if it's a little bit off, but it won't respond as vigorously unless the stimulus is directly in the receptive field in the orientation of this particular simple cell. Complex cells also respond best to correctly oriented bars of light, but they respond particularly to moving stimuli across the cell's receptive field. And in general, they tend to respond best to stimuli moving in a particular direction. So here we have this bar of light. The cell is responding a bit. When this light moves in this direction towards the left, we get maximal responding to this particular stimulus. When the stimulus is moving to the right, we get no responding at all. And so this particular kind of complex cell is designed to help us, our visual system, respond to movement. So as stimuli are moving across our visual field, we have cells that respond in particular to cells in a particular direction. So we get maximal responding to features in a particular orientation and in moving in a particular direction. So the simple cells respond just to a stimulus sitting there. Complex cells respond best to stimuli that are moving. End-stop cells respond best to moving lines of specific lengths, into corners and edges, and these are sometimes referred to as edge detectors. And this helps us determine where a stimulus starts and stops. And so these respond best if they don't have an entire visual field covered like we saw in the simple cells, rather where a stimulus ends within their visual field. So part of it's excited by illumination in one field and no illumination or less illumination in the other part of the field. And so this helps us to determine precisely where things start, stops, where they have corners, where they have edges, that sort of thing. So together, these cells are critical for processing specific features of a stimulus. So if you think about the screen in front of you, you have stimuli of particular orientations that end in particular places. So here we have a stimulus that's in this orientation and this orientation. To make this X, that's stopping in these particular locations. All of these features collectively respond to specific features in our environment. And this is how our perception begins to get built, is on this basic process of visual receptive fields with excitatory and inhibitory areas that start to respond to specific shapes in specific locations, moving in particular directions, and starting and stopping in particular locations. So based on this idea, we can start to understand how these features are driving our perception. And one of the ways we can do this is through selective adaptation. So by presenting specific stimuli, we can cause adaptation of only those features. And so then we can see sort of what our perception is after that. So if you remember, one of the things we've talked about is that our visual system will adapt out. And we'll talk in particular in more detail about this in our color vision section. If you look, for example, at a specific stimulus for long enough, your visual system will stop responding in the same way. And then when you look away from it, you'll actually see the opposite pattern. So if you stare at a yellow light, for example, and you look away, you'll see blue. Here in a moment, we're going to look at a motion detection convolution, and I'll send you off to some websites you can do to look for this particular illusion. So we can look at things like orientation gratings and contrast gratings. And so you can basically have someone look at one of these stimuli for a period of time, adapt out those features, and then see what features you can and cannot detect. So we can cause adaptation of those features presented, and that this indicates is there are cells responding just to these specific features. And so this helps us to provide further evidence for this idea that there are specific orientation gratings, orientation-tuned cells, contrast-tuned cells, etc. One of the things we can also look at is adaptation of our motion system. So this is called the counter-rotating spirals illusion. Let's see if I can get this one to actually work. Does not seem to want to work. Well, let me find another version. One moment. Here we are. Okay, so you can stare right into the center of this stimuli, and you can go, oh, sorry, it started for us already. So you can stare into the center of this stimuli, and it will adapt out your motion detectors for moving inwards. You can see all of these stimuli are moving inwards. And then if you do that for 30 seconds to a minute, and then look at something as you showed here, it will actually make them move outwards. You can take Buddha out of this. There are other versions of this same phenomenon. This is the spiral after effect. This one's particularly good. So I recommend going to this michaelbach.de website. You can just Google spiral after effect, and then you can actually play around with this, stare at the center of the screen for a while, and then look away. Looking at faces is particularly interesting because you can usually see them moving outwards. And what's happening while you're staring into the center of this counter-rotating spirals is you're adapting out those motion detectors. And so when you look away, things move in the opposite direction because the opponent process begins to take over. So this demonstrates we have cells in the visual cortex responding to these specific kinds of features and these specific kinds of stimuli. So the other thing I want to talk about when we're talking about feature detections and perception is I want to talk a little bit about contrast sensitivity. It's one of the ways we can determine how this kind of adaptation influences our perception. So if you have someone look at a contrast grading or an orientation grading, it might influence their contrast sensitivity. So contrast sensitivity we look at by measuring our contrast threshold, which is the percentage of contrast required to detect the presence of a grading. So 100% of our 100% contrast, is black, pure black on pure white. Then we can do black on gray or gray on white, and so we have reduced percentage contrast. The grading is described in terms of what's called cycles per degree of visual angle, so the number of times a cycle repeats in a spatial frame. And so a high degree of cycles per degree of visual angle is a lot of lines in a short space, whereas, say, one cycle per degree of visual angle is a wide grading. So we can then look at what's called the contrast sensitivity function. So it looks something like this. So where on the left we have shorter or smaller gradings, that is fewer cycles per degree of visual angle, whereas we get to the far right, you can see the grade gets closer and closer together. And then the further you move up the figure, the lower the contrast gets. And so you can probably see, depending on your particular visual abilities, I can see about to here. As you start to get older, our ability to discern contrast gets lower. And so what we see in older adults is reductions in that ability to discern contrast. So the amount of contrast required gets higher the older we get. So if you look at sensitivity, which is how well we can actually discern contrast, it gets worse and worse. You can see it gets lower and lower as we start to get older. So we can discern lower and lower levels of contrast, or we require higher levels of contrast in order to perceive those kind of higher spatial frequency stimuli. And so as we get older, it starts to get more difficult, particularly once we reach our 70s and 80s. Seems to be a major tipping point. You can see that's where there's a bit of a break in this area. So a little bit about how these feature detectors help us to see, and also a little bit about how it starts to change as we get older. So I want to turn next to talking about how we map out the visual cortex. We'll review quickly the lateral geniculate nucleus of the thalamus, and talk about its retinotopic mapping, and talk about how that mapping continues at the cortex, talk about orientation columns, and then ocular dominance columns to see how this is all put together. So the first thing, we have retinotopic mapping from the retina to the lateral geniculate nucleus in the thalamus. So we continue mapping the retina at the level of the LGN, which we talked about in the previous lecture. That mapping then continues to the cortex at specific locations. So we have location columns responsible for specific locations on the retina. Those are contained in this column on the cortex. So if we start looking at how this is all put together, if we go through vertically into a column, we might have a single location with varying kinds of stimuli. If we do an oblique electrode penetration, we get different orientations. So neurons within columns fire maxillary to the same orientation of a stimulus. The adjacent columns change their preference in an orderly fashion. So you can see we go from horizontal up to vertical, back around to horizontal again. And about 1 millimeter across the cortex represents an entire range of orientation. So in a tiny amount of cortex, we get this entire range of responding from horizontal to vertical, back to horizontal again. This demonstrates a really remarkable amount of processing power within the visual cortex. So these orientation columns help set up the way in which we're able to actually see particular stimuli. Now going up another level, we start to get ocular dominance columns. So we go out into a larger level, and so within each ocular dominance column will be other columns, so other orientation columns, for example. But then we go from left eye to right eye to left eye. So the neurons in the cortex respond preferentially to one eye. The neurons with the same preference are organized into columns. The columns alternate in a left-right pattern every 0.25 to 0.5 millimeters across the cortex. And this helps form the basis for stereopsis. So, I'm sorry I misspoke a moment ago. As we look across these columns, we see ocular dominance columns in particular layers. This is in layer 4. We also see that this alternates about every quarter to a half a millimeter, whereas it takes about a millimeter for the entire orientation site, the orientation stimuli to go through their full sets of orientation. These ocular dominance columns start to set up when we are very young. This forms our ability to see in depth. So stereopsis is how we primarily see in depth. Infants who are born with strabismus or amblyopia, which are cross-eyed or lazy-eyed, oftentimes do not set up these ocular dominance columns, and as a result have difficulty with stereopsis. It's possible to overcome that through later life training, but only a few people have accomplished that, certainly only one that I know of, and it certainly requires a lot of effort. But it's possible to actually overcome that process. Final note here is the what versus where pathways. First, there are many more visual processing areas after V1. We're not going to get into the full set of 2, 3, 4, and 5 and what they are all involved with. I do want to talk generally about the what pathway versus the where pathway. The what pathway is also known as the ventral stream. It originates in the parvocellular system in the LGN layer, which is, of course, layers 3, 4, 5, and 6. This is where we see fine detail. This is how we recognize faces. This is how we know what things are, hence its name, the what pathway. It's known as the ventral stream because it moves through the ventral part of the brain. So we can see we have the primary visual cortex here. So we have the striate cortex, which is the primary visual cortex, and then we start to move into the extrastriate cortex. And this is where we start getting differentiation of the what pathway down here into the temporal lobes and the where pathway up here into the parietal lobes. So we go from the eye to the LGN of the thalamus, primary visual cortex, and then up to these upper visual processing areas. The wear pathway is also known as the dorsal stream. It originates in the magnocellular system, so layers 1 and 2 of the LGN. The wear pathway is primarily involved in location information and motion information. So things that are moving are generally being processed by the wear pathway. We'll talk, when we talk about attention, towards the end of the semester, about how the visual attention system interacts with the wear pathway, particularly in the inferior parietal lobule. So essentially it goes this way. Starting at the retina, we have the scotopic and photopic systems. The scotopic system feeds into the M ganglion cells, to the magnocellular system in the LGN, and then moves up to the dorsal stream. Then we have the photopic system feeding into the parvocellular ganglion cells, the parvocellular system in the LGN, and then to the visual cortex, and then on up to, or on down to the ventral stream, or the what pathway. These two pathways interact with one another. We'll talk about some evidence for that in a moment. So, very quick summary of some of the neuropsychological evidence for these divergent pathways. We could do an entire semester on this topic alone. But some evidence for this idea of these divergent pathways are things like prosopagnosia. People with prosopagnosia lose their ability to recognize faces. There's a part of the brain known as the fusiform face area. They'll often have had stroke or some other sort of damage to this part of the brain, and they lose their ability to recognize faces. There's a related disorder known as agnosia, where they lose their ability to recognize objects. So those are the kinds of things that can damage the what pathway. So you can see the fusiform face area is down here in the mediotemporal occipital gyrus. So down here in the what pathway. Other neuropsych evidence for these divergent pathways are things like mirror neurons, which allow us to both perceive people's actions and engage in those actions. So we'll talk about this more when we get to the motor system. But there are neurons that respond to, say, looking at somebody's facial features and then creating those somebody's facial expression and creating those same facial expressions. The same neurons respond to both seeing that action and engaging in that action. And so we think this is part of our social functioning and observational learning. There are some rare cases of what's called akinetopsia. And these are individuals who have lost their ability to perceive motion, and this occurs in area MT, which is in that dorsal stream. Finally, just a quick note, we know that these pathways intersect with one another because of what we call biological motion perception. We can actually recognize objects, in particular people and animals, etc., based on what's called biological motion. And so we can actually look at an outline, say, of dots and know that that looks like a person. That is, we can determine what something is when it's moving. And so one of the things we know is that these pathways talk to one another because we can recognize what something is simply by the way it's moving. And oftentimes we can recognize people based on their gait without seeing their face. So that's a little bit about biological motion. This is sort of the end of our discussion of the cortical visual processing. The last bit is then to next look at color vision, which is kind of a whole separate discussion, which comes up next.